So we're live. So one of the things that I want to start with, I'm going to try to talk a little louder than I normally do, just so I can make sure I capture the audio pretty well. Is instead of starting off with something super sciencey, I thought I'd start off with something kind of fun. I don't know if this was this the episode you were telling me about, Jack?
It's one of them. They should talk.
Okay, so if any of you guys like watching Naturama at all, there is, my husband and I caught this the other night, is the most hilarious episode of Futurama that relates to this class. Like a lot of the examples they make fun of in this video, we actually use in this class and in other classes in our curriculum. So if you want a nice laugh, instead of me going all hardcore science on you guys first thing in the morning here at 8 o'clock, check out Naturama. It's an episode of Futurama, and it's really, really funny. So the first thing I want to ask you guys too is how did the online lecture for Thursday go? Did anybody have trouble with the online lecture at all? Was it clear, combined with hopefully your readings? And the University of Utah website is actually really, really nice. I really like that as a general website for teaching genetics. So no issues, no problems, concerns. Awesome. So you guys feel pretty confident about that material? Feeling good? Okay, good. All right. So so what I'm going to do is I'm going to start from the end of that lecture then, and then if you have questions, we can always go back and discuss anything that maybe you didn't quite get from that online video, but I'm going to kind of start from there. We will have a quiz this Thursday. Will be the last quiz before your exam. Okay, so this is the last quiz before your exam. We will do a mitosis laboratory activity on Thursday. It's kind of important to be here, unless you're really, really sick or something really important happens. We will be looking at cells on Thursday, and it's good for you guys to get hands-on experience with that stuff because you'll see photographs on the exam, and so you experiencing those live cells won't throw you off. If all you're used to is looking at cartoons, and then I throw a live example at you, unless you've had some experience with that live example, you can get a little confused, okay? So mitosis activity on Thursday. So we'll essentially have the quiz on Thursday and then jump right into the activity. I'll post the laboratory guidelines, the procedure for that activity after class today. So that if you want to review it before you come into class, it's a good idea. It's a part of your reading for Thursday. But it also help you go through the procedure really quickly. You see everyone?
Is the quiz going to cover like last week?
Yeah, so the quiz will cover the material that I covered on the lecture on Thursday, right? And then whatever I cover today. Okay, so nothing from Tuesday and previous Thursday, okay? Your quiz substituted for that. I'm hoping that you guys studied that material anyway, so you feel like you're kind of ready for that. Really, the purposes of the quiz, it's really just to give you opportunities to earn more points and to keep you up to date. So since I already have something great for you for Thursday, I think that's probably enough. I think that you covering the online material from Thursday and then the material from today is good enough. Good question. Any other questions? So as far as your reading is concerned, focus on mitosis and read the lab handout. Any other questions? Okay, our first exam I have put off by a day, and I decided to do that because of potential snow day today, so I may not even see my second section of class. Okay. But I also did this because we had a snow day last week, so there's been a lot of online stuff for you guys. So I want to make sure that I give you enough time to ask questions. Also, since we're doing the lab on Thursday, it's actually kind of nice to have another class right? Because that lab is full. I mean, we go the whole time during that lab. And so it's nice to have some extra time right before the exam just in case you guys have questions, or just in case I need to cover some additional material. So exam next Thursday, not this Thursday. That would make me nervous. And what I'm going to do is I'm going to post a study guide this Thursday. So I always try to post a study guide the week before. Some of you guys I've talked to already. I actually think that, study guide is nice that I put together. I think a smarter way to study for this exam is for you to put together your own study guide and then compare it to mine. I think that's way more effective than just reading my study guide over and over. So take a look at the study guide, which will be posted on Thursday. I think that's it before the exam. Are there any questions that you guys have before we get going? Okay, great. All right. So you guys were sort of exposed for the first time, well, not ever in your life, but for the first time in this class, you were exposed to the cell cycle. Okay? And so the cell cycle is a cycle that pretty much all cells undergo. And all cells undergo different phases of the cell cycle for different periods of time, even within your own body, but most certainly from organism to organism. But all organisms have a cell cycle, okay? And the three phases of the cell cycle are the first gap phase, the DNA synthesis phase, the second gap phase, and then some sort of cell division phase, whether it's mitosis or meiosis. What happens during the first gap phase? You guys tell me. Let's go ahead and start being a little bit active this morning, even though it's early. What happens during the first gap phase? You can look at your notes, you can look in your text, but you guys tell me what happens in that first gap phase. Sounds like Lucia. Yeah, so like general cell function, right? I mean, really, the first gap phase, a lot of your cells, in fact, most of your cells, are in the first gap phase, and they stay in the first gap phase for most of your life. Okay, this is basic cell functioning. The cell is undergoing transcription and translation. Okay, proteins are being produced, and then your cells are just, they're really, it's basic cell functioning. Okay. What happens in the synthesis phase of the cell cycle? Somebody other than Lucia who nailed it. It's where the DNA is copied, right? This is where DNA is replicated. Listen, unless a cell is getting ready to undergo a division, it doesn't go into synthesis. Okay? Once a cell goes into synthesis, it completes the rest of the cell cycle, period. Unless something goes wrong and the cell undergoes programmed cell death, okay, which we'll get to later. But once a cell enters the synthesis phase, this sucker is going to divide. Divide or die, essentially. Okay, so DNA is copied in the synthesis phase, and then what happens in the second gap phase of the cell cycle? So Lucia did a good job with the first one. Heather got synthesis, DNA replication. So what about that third phase? What's going on there? What about the third phase? You want to give us a stab? Yes, Madison. Yeah, it's getting ready to divide, right? So this cell, okay, that's been undergoing just general cell function, it's getting ready to divide, it copies its DNA, and there are other things that the cell needs to do to get ready to divide. The cell starts to produce organelles. So the cell starts to produce extra mitochondria. it's a plant cell, it starts producing extra chloroplasts. Okay? It starts producing proteins and enzymes for division. Okay? So yeah, exactly right, Madison. So second gap is preparation for division. And that involves replication of organelles and the production of the proteins necessary for division. Very good. And then of course, then you have your division phase, which today we're going to focus on mitosis, which is one type of division, in which those replicated copies of DNA are segregated into two cells, and from one cell you get two identical cells. Cells that are identical to the parent cell, okay, in genetic makeup and function. That is, unless something happens, right? So if you think back to the online lecture, when do most point mutations happen in this whole cell cycle? Relating this back to the cell cycle. When do point mutations happen? Nick?
DNA synthesis.
It happens during DNA synthesis most of the time. For the purposes of this course, certainly. The place where most point mutations happen is in DNA synthesis. Okay. Now the next question is, when are these mutations most commonly realized? In other words, where in the cell cycle do these mutations normally have the biggest impact? Where in the cell cycle? If the mutation happens here, okay, during synthesis, where else in the cell cycle, or where is the biggest impact to the cell cycle of those mutations? And make sure you link, try to make sure you link back these processes to the cell cycles. Go ahead, Maria. Right, okay, so Margaret's right. She says, well, any mutation here is going to be passed on to these daughter cells. And then what? You're right. You're totally right. But then what? And a mutation, if it's a gene that impacts the production of a protein for mitosis, right? Mutation happens there, certainly the impact can happen immediately, right? But more likely, where do you think that mutation is going to have an impact? The biggest impact. Pardon? The offspring? Well, that's mitosis, right? The offspring of the parent cell or the daughter cells. That's what Margaret said. One more extension of that. I'm sorry? Yeah, certainly. If it's a gene that impacts division, process of division. But most profoundly, what's the last possible guess as to the phase that's impacted? Mitosis, and we already talked about that. First gap. Yay! Yay! Very good. 3 choices, we finally get to first gap phase, right? So most of the time what happens is when a cell is impacted here, it can certainly, if the mutation impacts a gene that's important for division or preparation for division, of course it's going to impact the cell then. But once that mutation is passed on to the daughter cells and the cell starts general function, it's producing proteins for function, that's really where the impact happens. Okay, that's generally where you see the biggest impact of a mutation. But you can certainly see it in the second gap in mitosis. But sometimes the most profound impacts, those impacts that potentially impact variation in a population, it's right here in this first gap phase. If a mutation, this is sort of getting back to what you read on the University of Utah site. If a mutation happens during synthesis in a skin cell, okay, so if what mutation happens during replication in a skin cell, does it create variability upon which evolution by natural selection can act, and why? The mutation happens during synthesis in a skin cell. It can be my skin, plant skin, whatever. Does it create variability upon which evolution by natural selection can act, and why or why not? So Jack's answer is no. Okay, so why?
Because if it's happening in a cell that isn't a sex cell, it's not necessarily a heritable trait.
Exactly. Right? Postulates of evolution by natural selection, variability in a population. That variability or traits that might be impacted by natural selection have to be heritable. A mutation that happens in my skin cell, okay, I'm not going to pass down to the next generation. So those kinds of mutations are significant to individuals, perhaps survival of the individual, but even potentially reproduction. But the trait can't be passed down from parent to offspring. So no. Because remember when when you read the University of Utah article, it said really where mutations impact evolution by natural selection in sexually reproducing organisms, it's during the process of meiosis, right? Not mitosis. Very important that you make those logical links. How might a mutation that happens during synthesis, okay, forget the skin cell thing, just a mutation that happens during synthesis, impact organisms that reproduce asexually? Okay. How might these mutations impact organisms that reproduce asexually, that is, without meiosis? What is the impact? Is the impact bigger there? Can a mutation then impact evolution by natural selection in a population of organisms that reproduce without meiosis? Steph, want to take a stab? Don't ever make another organism, right? So the only way they reproduce themselves, I'm sorry to talk about. The only way they reproduce themselves, right, is by some process, some division like mitosis. If they're reproducing them by mitosis, any mutation that happens during synthesis is going to be passed down to the next generation, right? Everybody with me there? Okay, so yeah, the mutations that happen in organisms that reproduce asexually, that use mitosis as a form of reproduction, certainly these mutations here could have a very dramatic impact on the population after mitosis. Yeah.
To clarify, so asexual things, what are they, just like split in half?
Yeah. So most organisms on the planet do not reproduce with sex. They don't use sex to reproduce. Okay? They literally copy their DNA and then they have one cell that splits into two. Right? And that's the next generation. So most organisms on the planet don't reproduce by sex. And for those organisms, a mistake here has very profound effects on the next generation. That mistake is passed down and heritable. So yeah, it's a little bit different. I know you guys have always mostly focused on sexually reproducing organisms in your classes, but like I said, I'm going to emphasize organisms that don't reproduce by sex, because those populations are very dramatically impacted by these mutations. Bacteria, remember the bacteria example? Right? The mutation occurred, the bacteria divide, and it's immediately passed down. That's an example of an asexually reproducing organism. Okay, and that can have very, very profound effects on the population, as we saw with tuberculosis. Okay, there are lots of single-celled organisms that reproduce without sex that are eukaryotic organisms. Bacteria use binary fission, so they don't ever divide by mitosis, but it's a very similar process, and we'll talk about that soon. But even plants, which normally reproduce with sex, they can reproduce asexually. If your mom has ever taken a piece of a vine from a neighbor and put it in a glass of water, and you have roots form at the base of that plant has reproduced with itself without sex, right? Another individual has been reproduced from one. And that's reproduction asexual. Okay. Basically, if we kind of get back to talking about mitosis a little bit more specifically here. Okay. All cells have some sort of cell cycle and divide, right? So what we're going to do is, this cell, when this video started, was essentially right here. Okay, DNA had already been replicated, the cell had prepared itself for division, and now it's undergoing mitosis. And so what we're going to do is we're going to talk very, very specifically about this process. What exactly is happening during every stage of mitosis? This could be, this happens to be just a newt cell, but this could be any cell. Okay, so what happens during this process? What are the functions? In almost every class you guys have probably had since maybe junior high, the dadgum professor makes you go through the dadgum steps of mitosis. Like almost all of your biology classes at some point have had some sort of mitosis in it. Okay. The important question is, and one of these questions that I always ask my students is, why is it taught in every single class? Like, why is it that we can't just let this go? Why is it such a fundamental thing that we have to go on and on about it for at least an hour and 45 minutes and longer with examples and visuals? Why is mitosis important? We're going to focus on eukaryotic organisms here and cell division. But what are the functions of cell division in general? Why are they important? We just talked about one. What is it? Tell me why mitosis is important. Or just cell division in general. You guys got this. What's one example? You mean you've studied this in every single class, every single biology class you've ever had. Talks about mitosis. You don't know why. That's so lame. That's so lame. Okay, not you. It's lame that you don't know. Because you've been taught it every time, so certainly its function and importance should have been talked about. Reproduction, absolutely. Without cell division, you do not reproduce. This is particularly important for organisms who who don't use sex for reproduction. So without mitosis, you don't have reproduction in organisms that don't use sex. In sexually reproducing organisms, cell division, meiosis is important for the production of sperm and eggs, right? Got to have that happen in order for sexually reproducing organisms to do their thing, okay? But in particular, mitosis, which we're going to kind of come back to over and over again now, single-celled organisms rule the world. And the way that they reproduce and pass down their traits from generation to generation is very often by some sort of cell division that's mitosis or binary fission. Period. Absolutely crucial. That's why it's in every biology class. Another one is growth and development. The way that you go from a fertilized egg, a zygote, to what you are sitting in the seats right now is because specifically of mitosis and differential cell and cell differentiation. Okay, gene expression. That's why it's fundamental. Without mitosis, if you are a multicellular organism, you don't grow. Very, very important. Tissue renewal. You've ever had surgery? You've ever cut yourself and healed? You should be like, oh, thanks, mitosis. Okay, for not being a bloody dead mass somewhere. Okay, so it's mitosis that allows for tissue renewal. It allows for wound repair. So it's very, very important in most multicellular organisms. Okay, these are pictures of HeLa cells, which, by the way, if you haven't read The Immortal Life of Henrietta Lacks, you have got to read that book. It is just the most awesome book. It's about the woman whose cells were taken and reproduced. They were the first cells ever grown in culture and have contributed major breakthroughs in science. But it's a really, really interesting book. I encourage you guys to take a look at it. And this is a link to a really nice little NPR segment about the book and about this woman's life and the impact that these cells have had on science. So tissue renewal, absolutely. Absolutely. If you've cut yourself, thank you, mitosis. Okay. So, going back to the cell cycle, we all know how mitosis and meiosis, any kind of cell division is important. Reproduction, growth and development, tissue renewal. Okay. We're going to start in our discussion of mitosis here with DNA synthesis. Okay, so actually, excuse me, we're going to start before DNA synthesis. We're going to start right here in the G1 phase of the cell cycle. So this is a cell that is just about ready to go into synthesis. Within that cell, before the synthesis phase, each of the chromosomes is unreplicated. Okay, so here they're showing, here's this gene right here that we're going to follow. This could be the gene for anything, okay? These unreplicated chromosomes in the G1 phase are generally, you can't see them in the cell. Like when you look at cells, you can't see individual chromosomes. They're completely elongated at this stage. Okay. Even into synthesis, you have these very, very long chromosomes. If we're talking about a eukaryotic organism with linear chromosomes. During replication, what happens is that unreplicated chromosome is replicated during synthesis. Okay, so here you now have a replicated chromosome with two identical, if no mutations have happened, two identical copies of a gene. Okay, right before mitosis. Okay, the chromosomes are very long. Okay, they're very, very long. You cannot see them in the cell. But right before mitosis, the chromosomes condense. Okay, the chromosomes are composed of DNA plus proteins, okay, the histone proteins, and then what happens is right before mitosis, the chromosomes condense. Any pictures you've ever seen of a chromosome that aren't done with an electron micrograph, which is what this is, are always pictures of replicated chromosomes that are getting ready to undergo mitosis, because really that's the only time you can see them well. So anytime you've ever seen a picture of a chromosome, it's right here, getting ready to undergo mitosis, or it's in some phase of mitosis. Okay, so very long chromatin, and then right before mitosis, you get the chromosome becoming condensed. Here is that gene, and the now condensed chromosome. Here are the two copies of the chromosome. So each one of these these are sister chromatids. Okay, and these things are what are going to be separated during mitosis from one another. Everybody with me? Here are the basics of what we're going to take a look at today. If you look here, they're showing a cell in the G1 phase of the cell cycle. What's wrong with this picture is that the chromosomes are shown as condensed, right? Normally you wouldn't be able to see these. All of the G1 cells that I'm going to show you and I'm going to ask you to take through mitosis in this class are all going to be condensed, even though you know that in G1 the chromosomes are never condensed. Okay, they're still very, very long and you can't see them. But here's the G1 phase of the cell cycle. You have the synthesis phase where each one of these chromosomes is copied, and here are your sister chromatids. This is your parent cell that's going to undergo mitosis. The chromosomes condense, okay, and then each one of these sister chromatids is moved into a different cell. Okay, so from a parent cell, and then these chromosomes become uncondensed, okay, right after mitosis is over. So this is the process. So a really easy way to always know that you're looking at two daughter cells. If you have a picture of a parent cell and two daughter cells, the way that you know that that cell has undergone mitosis to produce those daughter cells is that the daughter cells are genetically identical to the parent cell. The cell before replication happened. Okay, so here you see 4 chromosomes, 4 and 4. Yeah, Jack.
So a chromatid is a replicated chromosome?
Correct. And then they're called sister chromatids because they're identical. Unless something crazy happens. Yeah. Unless something crazy happens, which I'm going to show you lots of crazy stuff happens, but right now we'll keep it simple. All right. So let's go through this. Everybody with me so far? Okay. Now we've gone from G1 phase of the cell cycle, general cell function. The cell moves into the synthesis phase. And remember, any cell that starts to move into the synthesis phase is always going to undergo some sort of division. Okay, so this cell has replicated all of its DNA. I'm going to talk about linear chromosomes here in this discussion. In the G2 phase of the cell cycle, this is interphase. A part of interphase, G1 is interphase, synthesis is interphase, and G2 is interphase. They are all subsets of interphase. Okay. This is the G2 phase of the cell cycle. They're showing the chromosomes are uncondensed. They're still long and linear. You really can't see the different chromosomes at this point. In this photograph, for example, all of the blue area will represent chromosomes. Okay. Any of the sort of greenish material, those are fibers. Okay, are microtubules. Okay. During the G2 phase of interphase, right before mitosis, this cell is getting ready to divide, right? So it's producing, if this is an animal cell, it's making lots of mitochondria. It's making all the proteins it needs for division, okay? And in this case, what's happening is it has replicated each of these centrosomes. So there was one centrosome, and during the G2 phase of the cell cycle in preparation for mitosis, in which organelles are being produced, 2 centrosomes are produced from one. Okay. Each centrosome contains 2 centrioles, and these are sort of the microtubule forming core here. Okay, so you have an intact nuclear envelope, you have chromosomes that are replicated but not condensed, and you have 2 chromosomes and all the organelles and proteins necessary for division. Okay, then during prophase, what happens is these centrosomes start producing microtubules. Okay. And these microtubules consist of individual tubulin units. Okay. And what these centrosomes are doing is they're producing microtubules. Okay. And the way that these centrosomes separate from one another is that these microtubules actually interact with each other. You can't see it here in this diagram, but those microtubules interact with one another and push each other apart. Okay, that's how the centrosomes sort of separate here. The nuclear envelope is intact. The chromosomes have started to condense. But this is where the formation of the spindle starts, is in prophase. Okay, here are the 2 centrosomes. You can see that they're producing microtubules. Those microtubules are going to be interacting with one another, pushing those centrosomes to opposite ends of the cell, essentially all the way along the periphery of the cell. The chromosomes are starting to condense. You can start to see chromosomes now where before you couldn't. In prometaphase, what happens is you have the centrosomes on opposite ends of the cell. Okay, they've been producing microtubules all along. See how they show the microtubules sort of interacting with one another. The nuclear envelope is starting to disintegrate. It's being broken down into individual units, which will then be put back together later. Okay. But these microtubules are starting to interact with the chromosomes. And any microtubule that interacts with the kinetochore, the kinetochores are these large protein units on either side of what's called the centromere. Okay, so here's the centromere that connects these 2 chromosomes, and the kinetochores are on either side of that centromere. And the microtubules produced by the centrosomes are interacting with these chromosomes at those kinetochore protein units, okay, on either side of the centromere, which connects the 2 sister chromatids. Okay. Then you also have microtubules that are not connecting to the chromosomes. These are called non-kinetochore microtubules. Your book calls them polar microtubules, okay, but they're another type of microtubule. In this case, what's happening is those microtubules are interacting with the chromosomes at the kinetochores on either side of the centromeres of the chromosomes. Okay, those are the kinetochore microtubules, and the other microtubules are interacting with each other, elongating the cell a little bit, enlarging the cell in some cases here. Okay. Again, important piece here is that nuclear envelope is breaking down. Unless the nuclear envelope breaks down, it's very difficult for the microtubules to interact with the chromosomes. Everybody with me? In metaphase, nuclear envelope is completely gone, completely broken down. The centrosomes have lined the chromosomes up in the center of the cell. Here are the centromeres. These little black dots represent the kinetochore protein units on either side of the centromeres. Here are those replicated chromosomes. Kinetochore microtubules, non-kinetochore microtubules, centrosomes. Okay. At this point, the chromosomes have all migrated to the center of the cell. And you can see that here. Here are the centrosomes. Okay. There are kinetochore and non-kinetochore microtubules here. No nuclear envelope. You can see here, you don't really see a nuclear envelope here either. Okay, it's just maybe a little bit farther along in prometaphase. In metaphase, again, what has happened is these sister chromatids are lined up in the center of the cell. Here are the polar microtubules or the non-kinetochore microtubules. Here are the kinetochore microtubules. These are the kinetochores on either side of the centromere. And then here's a little bit of the tubule. Okay. Now this is when the chromosomes at the kinetochore, the chromosomes move themselves to opposite ends of the cell. The microtubules, get this guys, the microtubules do not pull the chromosomes to opposite ends of the cell. The chromosomes, because of the kinetochore, which we'll talk about in a second, pull themselves to opposite ends of the cell. This is anaphase. Notice the cell's longer. It's longer for a reason, and it's because these polar microtubules or non-kinetochore microtubules are pushing against each other, elongating that cell. Okay. If you imagine each of these sister chromatids, I'm a sister chromatid, another of my sisters is heading that way, right? Microtubules connected to my kinetochore. Essentially what happens is the protein complex in the kinetochore drags me. It's almost like me grabbing onto a rope and pulling myself to off the end of the cell. And as I do that, the rope disappears. Okay? So I'm dragging myself to the opposite end of the cell, not being pulled, pulling myself. And as I pull myself, the rope is disintegrating. That's what's happening to the microtubule during anaphase. So these sister chromatids are being pulled are pulling themselves at those kinetochores towards opposite ends of the cell. This is what's happening here. Okay, this is the kinetochore. It's a protein complex. Here's a microtubule composed of individual tubulin subunits. So what happens is that literally these proteins grab onto that microtubule. Proteins associated with the kinetochore grab onto that microtubule. And as they do so, there are other parts of that kinetochore that pull those tubulin subunits off. Okay? So literally the sister chromatid is pulling itself to opposite ends of the cell thanks to the functioning of this kinetochore complex. The way they discovered this, somebody explain this to me. So here's what they did. If they to figure out whether the microtubules were being disintegrated at the centrosome versus at the kinetochore, they set up an experiment. And they applied fluorescent labels to the microtubules and to the chromosomes. And then what they did was they darkened a section of the microtubules. So they took away the fluorescent label for just a section of the microtubules. And what they found was what? What do you see here? This was a sort of discovery of how it is that sister chromatids move to the opposite ends of the cell, and whether the microtubules shorten at the chromatid end or the centrosome end. What would happen to this spot on the fluorescently labeled the darkened part of these microtubules. What would happen to this if the microtubules were shortening at the centrosome end? This would move, right? The darkened end would move towards the centrosomes, but it stays in place. That's how they originally figured out that something was going on at the chromatid end of the microtubule, and that indeed the centrosomes aren't pulling those those chromatids to opposite ends of the cell. Okay, it's a cool little experiment. Very simple way to test what's happening. Now, telophase and cytokinesis. Telophase, essentially what happens is the sister chromatids have been pulled to opposite ends of the cell. You can see that the cell is elongated, okay, from the pushing of those microtubules against each other in anaphase. The nuclear envelopes reform during telophase. Okay. The microtubules are just being broken down in the cell into individual tubulin units that are probably just going to be used for the next time the cell undergoes division. So in telophase, you have one cell, but two complete nuclei with reformed nuclear envelopes, and the chromosomes are starting to decondense. And notice you have only unreplicated chromosomes now in these individual nuclei. Excuse me. At this point, the cell can divide into two. And the actual division of the cell into two separate cells is called cytokinesis. Telophase means that the nuclear envelopes have reformed around each of those clusters of chromatids now. And then cytokinesis is when literally the cytoplasm is divided. And so from one cell, you now have two cells with complete nuclei. Notice that each daughter cell has only one centrosome, right? That centrosome will be replicated when this cell enters the G2 phase of the cell cycle and gets ready for dividing itself again. Okay, so one centrosome in each one of these cells, because the centrosome and that's the way it is, there's only one centrosome in any cell throughout the cell's life until G2, when the second centrosome is formed. There are a variety of different ways in which the cytoplasm can be divided in cells, or when they physically become two cells from one. And that is by what you most often see in animals is what's called a cleavage furrow. So during cytokinesis, what happens is actin and myosin, which are sort of the same proteins important in muscle, in flexing a muscle, literally squeeze these two cells apart. It's almost like taking a string and wrapping it around a balloon and then pulling the string tight. Okay, that's what happens during cytokinesis in animals. In plants, it's a little different, because plants have really, really hard cell walls. And so what happens is they literally, using the Golgi apparatus and vesicles, they literally lay down cell wall material between the two cells. So there's no pinching of the cell into in plant cells. Literally, a new wall and plasma membrane and cell wall is built between two cells in plants. It's a little bit different in these guys. So when you look at cytokinesis in a living plant cell, which is what you guys are going to see on Thursday, you'll know it's cytokinesis because you'll be able to see a little bit of a line here that's the cell plate. If we were looking at animal cells, you would see them pinching like a balloon into. Okay, there are lots of structures involved in mitosis. It's easy to get them confused, so practice using them. So chromosome, centromere, centromere, all sound very, very similar to one another. So make sure you practice using them. What's the difference between a chromatid and sister chromatids? What is the difference between a centromere and a kinetochore? The microtubule organizing centers. Okay, in plants, there are no centrosomes. So in some cells, there aren't centrosomes. There's these microtubule organizing centers. That's the only reason why they really pretty much use this. And then centrosome and then centrioles. And plants don't have centrioles either. So no centrosomes or centrioles. But it's important to practice using these terms so that you don't get them confused when you try to explain mitosis. So if we go back to the real thing now, so we're in prometaphase, right? The microtubules are interacting with the chromosomes and moving them to the center of the cell. Watch what happens to the size of the cell as the cell goes through this process. It gets bigger and bigger. This is right about metaphase. All of those cells are pretty much right in the center of the cell. There's anaphase, where the chromatids are pulling themselves to opposite ends of the cell. See how much longer the cell is? And then boom, you have cytokinesis. Animal cell, right? Because you can see the pinch. Did you guys see the pinch? Okay, so yeah, so that's the real thing. Here's what's going to happen on an exam for you guys. What I'm going to do is I'm going to give you a cell that I'm going to say is in the G1 phase of the cell cycle. Now, what's wrong with this again is that you can see the chromosomes, right? Normally in G1 you would not be able to see these chromosomes. Okay. I want you to, when you draw a cell undergoing mitosis, I want you to always draw the chromosomes condensed even though you know they're not. And it's only because I can see that you understand what's happening during the process. So what I want you to do is you can work on your own or in pairs. And I want you to take this cell through mitosis. And here's what I want you to do. Grab a marker. Okay, so you're going to start off with a cell in the G1 phase of the cell cycle. I want you to show me what that cell will look like at synthesis. Then what I want you to do is I want you to show me what it will look like in metaphase of mitosis, right? So we're skipping some pieces, right? So along this little line, you should be able to describe what's happening, okay, to get to metaphase. And then after metaphase, I want you just to show me what the cell looks like after cytokinesis. Again, it's really good to practice process. So describe what is happening as much as you can in these diagrams. Now, you're also going to want to label everything, right? So you're going to want to label chromatids. You're going to want to label centrosomes. You're going to want to label kinetochore microtubules. You're going to want to label polar or non-kinetochore microtubules. You're going to want to label the nuclear envelope. You're going to want to label the centromere. That's it. Those are the things that you're going to want to make sure to label in each of your diagrams. Okay. The best way to draw these is not to draw your cells this big. You cannot see what's going on in that cell. Draw a cell. I mean, when you draw a cell on your page, draw a cell. Draw a big cell so that you have room to label everything. Remember, you have to think about the cell cycle here because you're in G1. Okay, the way people mess this up is they don't do what? Because what has to happen between G1 and mitosis? What has to happen between G1 and mitosis? DNA has to be replicated. All right, people always, this is like the place where people mess up. If you remember to replicate your chromosomes so that you have two sister chromatids connected by a centromere, man, it's hard to mess up mitosis from there because then you always know that the daughter cell looks identical to the parent cell. Everybody see what I'm talking about? That everybody messes up. So draw these things, and then as a group, we're going to draw the cells on the board. I'm going to have you guys come up and do it. So let's work on this for a little while and make sure we get this done.